Right, today we are going to talk about glycolysis, right? And glycolysis, how do you define glycolysis? Glycolysis is a metabolic pathway in which you break down the glucose into two molecules of pyruvate. So basically this pathway is you break down the glucose through multiple steps, right? Eventually into pyruvate. And during this process, when you are breaking the glucose into two molecules of pyruvate, some energy is released, right? Some energy is captured as ATP and some energy is captured in the form of NADH, right? Now again, what is glycolysis? Glycolysis is a metabolic pathway by which we break down the glucose into two molecules of pyruvate or pyruvic acid. And during this process, there is some form of formation of ATP. There is modest amount of energy released, not big amount of energy, because during glycolysis, you release only a small amount of energy from glucose in the form of ATP and in form of NADH, and NADH later on can be taken to the electron transport chain, and there from this we make more ATP. And pyruvate, if mitochondria are available in the cell and if oxygen is available in the cell, then eventually pyruvate will enter into Krebs cycle or citric acid cycle, and lot of energy will be released. But when we talk about glycolysis, that is the pathway from glucose up to pyruvate, right? And if oxygen is not available, or mitochondria are not present in a cell, then we cannot take pyruvate into acetyl-CoA, and eventually we cannot take pyruvate into Krebs cycle. Then we take pyruvate into which side? Yes, lactic acid. Is that right? So it means that during the glycolysis, right, if oxygen is available, if mitochondria are available, the end product of glycolysis, which is called pyruvate, will be taken into Krebs cycle and lot of energy is released. And this type of glycolysis, right, in which pyruvate is formed in the end, is called aerobic glycolysis. But if oxygen is not there or mitochondria are not there, then pyruvate is converted into lactic acid, and when pathway moves into this direction, we call it anaerobic glycolysis. That is called anaerobic glycolysis. This glycolysis, the breakdown of glucose into pyruvate, this takes place in which cells? Jill. This reaction occurs in which cells? Gary. All cells. All the tissues have a capability to break down the glucose into pyruvic acid or pyruvate. Is that right? And most of the tissue have also capability to break down, to advance the pyruvate into acetyl-CoA, and most of the tissues will eventually take it to Krebs cycle if oxygen is available and mitochondria are there. But is there any cell which does not have mitochondria? Listen my question carefully. Is there any type of cells in your body which do not have mitochondria? And even if oxygen is there, because mitochondria are not there, those cells cannot take this into Krebs cycle. Those cells have to take the pyruvate to lactate. Again listen, my question is there any type of cells in the body in which mitochondria are not there, and even if oxygen is there, pyruvate, due to absence of the mitochondria, we cannot do the aerobic glycolysis, so we have to move to the lactic acid. Yes, Samad. Which cells? Red blood cells. Excellent. So Samad knew the answer that red blood cells right? They have what type of glycolysis? Anaerobic glycolysis. Even though red blood cells have a lot of oxygen. You know, red blood cells have hemoglobin and hemoglobin has oxygen. But red blood cells themselves don't utilize oxygen. The good news is that they are very honestly transporting oxygen from the lungs to the tissue, and for their own metabolism, they don't utilize oxygen. They are very honest transporters. They are very honest transporter of oxygen, because red blood cells don't have mitochondria, even though they have oxygen attached with the hemoglobin, but because they don't have mitochondria, so in red blood cells, pyruvates are converted into lactates. Am I clear? Now another thing, I told you that glycolytic pathway operates in every cell. Most of the enzymes, or rather the enzymatic pathway, or multiple enzymes which are involved into this pathway, these enzymes are present in which part of the cell? Yes, please. Madam Laura. She is going to tell us that which part of the cell has all the enzymes available for the glycolysis. In the cytosol. That's great. So in the cytosol, you can break down glucose into pyruvic acid, because there are multiple enzymes involved in this pathway, and all those enzymes are present in cytosol. So we can say glycolytic pathway or glycolysis is a cytosolic pathway, right? This is a cytosolic pathway. Now, any cell which is going to do glycolysis, what will be the first step? Any cell which is going to be involved in the process of glycolysis, any cell which want to get energy by the breakdown of glucose into pyruvate, what will be the first step? Okay, Sadaf is telling us. What should be the first step? What is the first step? Yes, Patrick.
To get a glucose and add the water.
Yeah, Patrick knows these things well. He knows first of all that cell has to get the glucose. Is that right? It is the same thing if I ask you, you are planning for shopping. What is the first step? To get the money in your pocket. Is that right? In the same way, when cells are going to do glycolysis, when they are going to break down the glucose, the step number one is they should get the glucose. Is that right? And cells are of course getting glucose from extracellular environment and from the blood, right? So the first step when we will talk about is that how glucose is transported from the blood and extracellular environment into the cell. Once glucose is transported inside the cell, then truly glycolytic pathway starts. So first we will talk about transport of glucose. When we talk about the transport of glucose, all of you must be knowing that glucose is primarily coming from, yes, from our diet, breakdown of the carbohydrates, right, monosaccharides, and eventually from here it is coming to the portal, which circulation? Portal circulation, and portal circulation is going to, yes please, liver, right? So glucose is coming from GIT, it is being absorbed through the portal circulation, it is coming to the liver, and in the liver there are hepatocytes, right? There are hepatocytes, the cells of the liver, right? And eventually blood is entering into liver as portal. What is this? Portal vein passing through the liver areas, right? And these vascular channels are surrounded by liver cells, and eventually this blood will go out of the liver, and now it is going to general circulation. It is going to, yes please, general circulation. Now this point is very important to understand this particular pathway, because when you take a heavy meal, especially the meal rich in carbohydrates, very large amount of glucose is absorbed. And it's very important to understand that when a very big amount of glucose is absorbed from the GIT into portal circulation, liver cells are specialized in extracting the glucose, right? Liver cells are specialized, I will talk later how, they will take up lot of glucose, right, and store it, and only a small amount of glucose will go to the general circulation. What is the benefit of this? The benefit of this strategy is that lot of glucose which is coming right out of it, most of it is taken up by hepatocytes or cells of the liver, so that after the meal, amount of glucose which is reaching to the blood, it does not produce very high level of glucose, right? So it does not produce hyperglycemia. Is that right? And of course, when in between the meals, when glucose is no more coming from here, is that right? When your GIT is not absorbing glucose before the meals, you can say, when you are in a fasting state, right? At that very time, amount of glucose here is very less. And later on in biochemistry, you will learn then liver cells are capable of releasing the glucose back to circulation. An advantage of this situation is that in the fasting state, blood glucose level does not drop dangerously. So how the liver is working? Liver is working as the glucose bank, right? When there is extra glucose, liver cells will take up the glucose, and when there is deficient glucose, they will release the glucose. But one point which is very important to understand, that most of the monosaccharides are glucose. What other monosaccharides are absorbed? Who will tell me? Yes, fructose and galactose. So it means from GIT, lot of glucose is going, some fructose and some galactose is going. The important point which you have to understand, the liver cells are master in special function, that liver cells can take up the fructose, they can take up the galactose and convert them into glucose and release the glucose back. Liver cells can take up, number one, they can take up the glucose and store it, and they will release the glucose when blood glucose level is falling. Number two, liver cells can take up the fructose as well as galactose and convert that into glucose. So in the end, the major fuel, major monosaccharide which is reaching to the tissue is glucose. Is that right? Even though from GIT some fructose is also coming, galactose is coming, but because liver is present between the GIT and the general circulation, and liver has a capability, liver cells have a capability to convert fructose and galactose into glucose. Is that right? Liver cells can take up the fructose, they can take up the galactose and convert into glucose, and they can release that glucose. So what we have seen, the liver cells can release the stored glucose whenever glucose levels are going down. Liver cells can release stored glucose as well as they can release that glucose which has been made by the conversion of fructose and galactose. Is that right? So it means that most of your tissues, right, let's suppose we have a cell here. This is a cell here. Most of the tissues in their extracellular environment. Extracellular environment means here it is interstitial fluid, and this is blood, right? Now, what is the difference of glucose concentration in the blood and glucose concentration in interstitial fluid? Someone with good knowledge of biochemistry will answer. First, listen to the question. My question is that of course blood is glucose is coming into blood. From there it should go to interstitial fluid, the fluid which is in between the cells, and from there the glucose should shift to the cells. Is that right? My question is, the glucose concentration in the blood and glucose concentration in interstitial fluid, what is the difference in that concentration? Question goes to someone who looks very intelligent, or talk.
The glucose in the blood can be changed.
Yes, I think Laura wants to say something. Yes, my question is, what is the difference in concentration of glucose in the blood and glucose concentration in interstitial fluid? Where is higher? In the blood is higher or interstitial fluid? People who believe that concentration of glucose in the blood is higher and concentration of glucose in interstitial fluid is little less, they should raise their hands. I want to see their beautiful faces. Okay, these are guys. And people who believe concentration of glucose is higher in the interstitial fluid, they should raise their hands. Right. These people are wrong. And who are these people who raise their hands? Yes, I want to see that. These are also wrong. The reason being, listen carefully, the reason being glucose freely shift between these two areas. We don't need, this is the first lesson, we don't need any transporter to shift the glucose from the blood to interstitial fluid. Is that right? Right? No, now you, what you are saying, please.
No, I'm asking about the concentration. It needs to be different, right?
Not significantly, because as cells take up the glucose, a little fall is there, glucose will immediately shift. Right? Because, listen carefully, the transport of glucose from the blood to interstitial fluid and from the interstitial to blood does not require any transport mechanism. Glucose can easily diffuse across the capillary membranes. That is the point to remember, right? Because glucose can easily pass through the capillary membranes. So, whenever we talk about the glucose concentration in the blood, that is what we measure in the blood samples of the patient. That is what we measure. Right? Whenever we talk about glucose concentration in the blood, actually the same concentration is present in for practical purposes, same concentration of glucose is present in interstitial fluid. Now, another question. What is the difference, the glucose concentration in interstitial fluid and glucose concentration inside the cell? Again, glucose concentration in extracellular fluid and glucose concentration in intracellular fluid. Right. They are usually equal or they are not equal? Who will tell me? Yeah, that's very important to understand that glucose, if someone make like that, the glucose can easily jump in and jump out. That concept is wrong. From here, it jumps around like young people. But here, it cannot jump around. Why it cannot jump around? The reason being that cell membranes are not leaky to glucose. Is that right? Glucose is a larger molecule and cell membranes allow smaller molecule to diffuse in and out. Because glucose is larger molecule, so it cannot freely enter into cell. It means when cells need glucose, glucose has to be transferred from the extracellular environment to the intracellular environment through special type of transporters. We have to have special mechanisms to get the glucose into the cell. Is that right? Now here we are going to discuss glucose transport mechanism from extracellular environment to the intracellular environment. Now, there are two types of glucose transport. Glucose transport across the cell membranes. There are two types of mechanism. One is facilitated diffusion. Facilitated diffusion. And other is, other is energy dependent mechanism, right? That is energy dependent mechanism, and usually their transport of glucose is coupled with sodium. You know, in some cells, glucose stick with the sodium and then sodium drag the glucose in. We'll talk in detail how. So we said, so glucose, sodium, yes, what type of transport? Co-transport, as you have co-education, transport. Right, so I have to explain, number one, how the glucose is going into cells by simple facilitated diffusion. And I have to explain to you that in some situations, glucose get into cell along with the sodium. Right, right now I will explain first of all facilitated diffusion. Right, what is the mechanism of facilitated diffusion? Let's suppose this is a cell and it wants to take up the glucose. As I told you that glucose cannot pass through the membrane. Is that right? Now we need some transport mechanism. So what really happens in the cells, of course you know there are nucleus, and in the nucleus there are genes, DNA, and some genes they express and make messenger RNA, and when that messenger RNA is translated into proteins. Those proteins will go into and get planted into membrane, and those proteins are like this. This is a special type of protein, and you know how many, now this special type of protein which is being expressed right on the membrane, this is transmembrane protein because it is through and through. Is that right? Now this protein is very interesting. What really happens that outside it has its open mouth, and from this mouth it will capture glucose. When this protein is expressed right all the time, it is like, it's just like your mouth, you know. When you don't have food, you keep your mouth open, but as soon as you get in, you close it. Is that right? So until there is no glucose here, this mouth remain open. As soon as glucose gets stuck here and here, what will happen? They will close this side and open from other side, and they will open their mouth from this side, and glucose will shift now inside. So what is happening? That this is a protein which is, this is a transmembrane protein which is through the membrane, and this protein has a capability to alter its shape. When it is not loaded with glucose, look at it, this protein is like this. Now it is not loaded with glucose. As soon as glucose sits here, it opens like that. Is that right? Now, this is the basic way how these glucose transporters work. Is that right? So, such type of proteins are called glucose transporters, and we call it glucose transporters, or from glucose and T for transporters, we simply call it GLUT, G-L-U-T, GLUT. These glucose transporters, there are up to now, they have discovered about 14 different type of glucose transporters. There are many types of glucose transporters which are expressed in different tissues for different functions. Is that right? But first of all, you must know the glucose transporters are proteins. Once they are expressed on the cell membrane, they bind the extracellular glucose and they shift the glucose to intracellular environment. And if glucose binds from inside, they will shift it to outside also. Is that right? Now, these glucose transporters, I told you there are many types of glucose transporters, about 14 different types of glucose transporters. Is that right? Now, different types of glucose transporters work in a different way. But a very important point which you must know about these glucose transporters is that these glucose transporters are acting as simple. Do you think these glucose transporters are using energy to transfer the glucose? They are not using energy. They are not using any energy, right? What they are doing, they are making the membrane permeable to glucose. When membrane has more glucose transporters, membrane becomes more permeable to glucose. When membrane has less glucose transporter, membrane is less permeable to glucose. So glucose transporters presence only determines the permeability of the membrane to glucose. Is that right? But how much glucose will pass through this area? It depends on the concentration difference of glucose. If glucose concentration outside is very high, then more glucose will be going in. If glucose concentration is very low, less glucose will be going in. So it means that basically glucose is moving from high concentration to low concentration. When any molecule is moving from high concentration to low concentration, we call it diffusion. What we call it? Process is called diffusion. So basically glucose is diffusing from high extracellular concentration to low intracellular concentration. But this diffusion is not simple diffusion. But this diffusion of glucose is not simple diffusion, because if transporters are not there, this diffusion will not occur. So this glucose diffusion is facilitated by the presence of glucose transporters. Is that right? So this type of transport of glucose is called facilitated diffusion. Why we call it facilitated diffusion? First we should know why we call it diffusion, because glucose is moving from high concentration to low concentration, so it is diffusion. But because this diffusion cannot occur simply through the cell membrane, so it is not simple diffusion. This diffusion has to be facilitated by the presence of glucose transporters. Am I clear? Now, there are many types of, different types of glucose transporters. For example, there is type 1 glucose transporters. Some of them glucose transporters you are supposed to know. Type 1, type 2, 3, 4, 5 and 7. These are the minimum types of glucose transporters which you are supposed to know. Now, type 1 glucose transporter and type 3 glucose transporters, they are present on most of the tissues. Type 1 and type 1 glucose transporters plus type 3 glucose transporters, they are present on most of the tissues, especially they are present on central nervous system and RBCs. Now, these transporters are expressed on many cells in the body. The purpose of these transporters is the basal uptake of glucose. Basal uptake of glucose mean that when type 1 and type 3 transporters are present over there, right, they allow the glucose transport into the cell under basal circumstances. Basal circumstances mean when glucose level is low. Is that right? Still glucose is moving to the cell. But if glucose level in the blood becomes very high, then some cells will need additional transporters. Some cells will need additional transporters. So basal transporters are glucose transporter 1, glucose transporter 3, and out of them, especially RBCs and brain, right, they are having much of these transporters. And these transporters are responsible, okay, I tell you something. These transporters, we say, they have high affinity. These type 1 and type 3 are having high affinity. When I say they have high affinity, it means these transporters bind loosely with glucose or tightly with glucose. They bind strongly with glucose. It means they can transport the glucose because they bind with glucose powerfully. It means they can capture the glucose and transfer the glucose even when blood glucose levels are low. Is that right? When blood glucose levels are low. These type 1 and type 3 glucose transporters are working. Is that right? So, now we come to some other type of transporters. Let's suppose we talk about type 4. Type 4 transporters, type 4 glucose transporters are mainly expressed on the skeletal muscles and on adipose tissue, right? Skeletal muscles and what are these cells? Loaded with the fat droplets, adipose cells. They have their cells have special type of additional glucose transporters, and these additional glucose transporters are, their concentration on the membrane is regulated by very important substance. Who will tell me? Insulin. You know, when your glucose level is going up, then beta cells of the pancreas will release insulin. Insulin will come and bind with cells of the muscles and bind with adipocytes and lead to increased expression of glucose transporter 4, so that more glucose should be transferred to the muscle cells as well as to the adipose cells. Let me tell you how it happens. Let's suppose this is a cell, and normally type 1 and type 3 transporters are there, and they take only very basal level of glucose N. But if glucose level in the blood, whenever glucose level in the blood become very high, what really happens? Insulin level also goes high. So insulin also goes high, insulin is released by the beta cells of the pancreas in response to increasing level of glucose. Is that right? So that insulin helps the cells to deal with the extra amount of glucose. Now what insulin is doing? Insulin has its own receptors. This is the receptor of insulin, right? When insulin bind here, when insulin will bind here with the receptor, what happens? These receptors give intracellular signals. Some of these signals, insulin stimulated receptor, and these receptors give intracellular signals. Some of these signals go to special type of vesicles. These are intracellular membrane bound vesicles, and in the wall of these vesicles there are special proteins. And what are these proteins? Yes, who will tell me? Insulin goes to adipocytes and to the muscle cells. Insulin binds with its receptors and it gives signal inside the cell, and once insulin signal come here, the result is that these will float towards the membrane, and this membrane, let's suppose this transporter has come here, this membrane vesicle is here, this part of the membrane and this part of the membrane, they fuse with each other. And what are these proteins? You are not telling me. These are glucose transporter 4. So what really happens, that fat cells and muscle cells have stores of transporters. They have stores of transporters in the vesicular membranes, and these vesicles are initially present within the cell. When insulin stimulates the receptor gives the signals, and these vesicles start moving towards the membrane. Then what really happens? Cell membrane and vesicular membrane fuse with each other. When they fuse with each other, the result will be that you will find this vesicle is fused with the membrane, and now vesicular membrane, which is loaded with transporter, this vesicular membrane become the part of the cell membrane, and then you find that expression of glucose transporter type 4 on the cell membrane surface is increased. This is what one of the function of insulin. Insulin has many functions. Again, it's worth repeating that when in your blood glucose level is going up, if your pancreas is normally function, beta cells of the pancreas will release insulin, and this insulin will come to the skeletal muscle cells as well as to the adipocytes, and insulin will bind with these cells here, and insulin will give, through its receptor, insulin will give intracellular signals. Some of the intracellular signals will activate certain proteins, and those proteins will pull the vesicles loaded with glucose transporter 4, and those proteins will drag these vesicles towards the cell membrane, and eventually vesicular membrane and cell membranes fuse with each other, and in this way glucose transporters which were previously present in vesicular membrane under the influence of insulin, now they are present in cell membrane, and of course permeability of the cell to the glucose is increased. So glucose will now more readily go into these cells. Is that right? So we can say the glucose transporter type 4 are, their expression in the membrane is insulin dependent, but these others are not insulin dependent. Am I clear? No problem? Right. So we were talking about type 4. Yes, you have a question.
That's an insulin receptor that is binding to an insulin.
Yeah. Look, insulin will come. It is just like that. Look, just a minute. Insulin is a hormone, all of you know, right? The hormone is released by the beta cells of the pancreas when blood glucose level is high. When insulin come to the blood, right, it will bind with those cells. Insulin will bind with those cells which cells are expressing the receptors for insulin.
Yes.
Is that right? Insulin is a very large molecule, it cannot go into cell. So insulin will just bind with the receptor and trigger the bell. It will give intracellular signals, and these signals do many things. When I will teach you insulin, I will tell you. One of the function is the signals will transport the vesicles loaded with glucose transporter 4 to the cell surface, and what will happen? The glucose uptake by these cells will be increased. There's a question. Neurons, brain cells, neurons use a lot of glucose, So should the neurons have type 4 glucose transporters or not? No. Why they should not have?
You need a base barrier.
Again, my question is that the neurons really need a lot of glucose for their energy. They produce a lot of ATP, and those ATPs in the neurons are specially used to run their sodium potassium ATPases, right? There are special cell membrane transporters present in neurons. Now, do you think neurons should have glucose transporters type 4 which are dependent on insulin or not? Yes, they should not have, because neurons are so important to us that glucose transport to the neurons should not be dependent on insulin. Is that right? The glucose transport. So neurons should have what type of transporter? They should have very high concentration of 1 and 3. 1 and 3. So in some books, that is why it is written that 1 and 3 are present on neurons. But actually 1 and 3 are present in most of the tissue, but these transporters are in very high concentration on neurons. So that this is very important organ. A brain for most of us is very important. Is that right? So central nervous system should keep on getting the supply of glucose under all circumstances. Right? Yeah? At the basal rate. Actually what happens? If neuron is metabolically more active, it will utilize the glucose more, so more glucose will go in. Because transfer of glucose through the transporter 1 and transporter 3 depends on the concentration difference of glucose outside and inside. If neuron become more metabolically active, it will use the glucose more, inside glucose level will become less, more glucose will shift in. Is that right? But muscles and what is this? Adipose cells, they can store glucose. Muscles can synthesize from glucose glycogen and store it. And fat cells can convert glucose into glycerol, which can be utilized in formation of triglycerides. That is why when there is excessive extra amount of glucose in our blood, insulin is there to specially push this extra glucose into muscle cells and into fat cells. Am I clear? So this is very special point related about glucose transporter number 4, which are dependent on insulin. Now there is another transporter that is truly a two-way transporter. There is another glucose transporter, and that transporter allows the glucose in two-way movement. For example, if glucose is high in the blood, there is a special type of transporters, and if glucose is high in the blood, glucose will move in this direction. And if glucose is high in the cell, glucose will move in reverse direction. These are two-way transporters. This two-way transporter should be called transporter number 1, 2, 3, 4, 5 or 7. Use your brains. Some glucose must be going there, I'm sure. There are two-way transporters.
2, 5 and 7.
Out of these types, which are two-way transporters? Use your brains. Of course, 2. Two-way transporters are glucose transporters type 2. Is it difficult to remember? It's so easy. I don't know why people memorize it. That some glucose transporters are two-way transporters, so they are fortunately labeled as glucose transporter type 2 for two-way. Is that right? Not difficult. Now there's the real question. You have to use your higher layers of your neurons. Which type of tissues or cells should have glucose transporters 2? just a minute. Let me make my question clear. I'm asking you a question that out of my all body tissues, which tissues and cells must have glucose transporters type 2? Yes, please. Yes, Shan. Liver. Why liver cells? Yes, you said it. You are going to tell me why.
Because it stores glucogen, it needs to release to be used as...
Excellent. So Patrick knows that liver is very important in one function. As you know, between the GIT and between the general circulation, what is there? They are liver cells, right? So glucose from the GIT, and this is general circulation. Now, as we discussed previously, that liver cells, when glucose concentration is high, when more glucose is coming from GIT, glucose should go into liver cells. And when no glucose coming from GIT and tissues have used the glucose and glucose level is very low, then liver cell should release glucose. So it means liver cells should have what type of glucose transporters which work two-way? Number 2. Is that right? Because liver cells are very special cells that when glucose level in the blood is high, they will take up the glucose and store the glucose as glycogen. And when blood level of glucose is less, liver cells can break down the glycogen back into glucose and release the glucose. Even liver cells can synthesize glucose, new glucose. That process is called gluconeogenesis. Let me explain it. Let's suppose here is a liver cell. When blood glucose level is high, glucose will enter into this cell and this cell will make the polymers of glucose. And these polymers of glucose are called, this is called glycogen. So liver cells can take up the glucose and convert into glycogen. This process is called glycogenesis, formation of glycogen. This is one function of the liver. When blood glucose level is high, it will take the glucose and make the polymers of glucose, store the glucose. Is that right? And when glucose level in the blood is less, then they will break down the glucose and bring it back to blood. So this process in which glycogen is broken, this process is called glycogenolysis. So actually there is a transporter here. This is glucose transporter 2. When blood glucose level is high, glucose is going to this process. Glucose is going like this. And when blood glucose level drop, glucose is going from here to out. So glucose transporters which are type 2, they should be present in which cells? Liver cells, which is our glucose bank. That when glucose is more, we deposit glucose there. And when we are deficient in glucose, we get the glucose from there. This process in which glycogen is broken down into glucose is called glycogenolysis. Don't call it glycolysis. That is not glycolysis. Glycolysis means breakdown of glucose into pyruvic acid. That is glycolysis. This is not breakdown of glucose. This is breakdown of glycogen back into glucose and glucose coming out. Okay. There is another process which is called gluconeogenesis. And by this process, liver cells can also produce glucose and glucose will come out. It means that when your blood glucose level drop, liver will donate you glucose. From where the liver cells bring glucose for you? Number 1, the breakdown of glycogen. The process is called glycogenolysis. Then they can make additional glucose from another process called gluconeogenesis. What is gluconeogenesis? Yes. Dr. Afia, what is gluconeogenesis? Anyone from here? Have you heard the word gluconeogenesis? What is gluconeogenesis? Yeah? Synthesis of glucose. But how it is different from glycogenolysis? Glycogen. Right. Okay, someone will make a better definition? Okay, the simplest definition is, there are many definitions. For a while we have a simple definition. Gluconeo, the stress is on the neo. Gluconeogenesis, glucose formation from new sources. New sources are carbon skeleton of fatty acids and carbon skeletons from amino acids. So we can say when fatty acids, carbon skeleton can be converted into glucose, or when from some amino acids, some carbon skeletons can be converted into glucose. We say glucose is being synthesized from new sources. Is that right? Gluconeogenesis. This is another simple way to tell what is gluconeogenesis. Formation of glucose from non-carbohydrate sources. Formation of glucose from non-carbohydrate sources. That's so simple. That is gluconeogenesis. Now, the point which I wanted to tell you, all organs which can do gluconeogenesis must have two-way transporters. So they should have glucose transporter 2. One organ which is involved in gluconeogenesis is liver. Any other organ you know which can synthesize the glucose from non-carbohydrate sources? I'm about to be impressed by someone. Again, listen. We were talking about glucose transporters, but we are trying to make some sense, right? Sense is that type 2 transporters should be present in those tissues especially in which glucose need to be transported in both direction, right? For example, one is liver. What is the other organ in which gluconeogenesis is possible? Because the second organ can do gluconeogenesis, then it should also be able to pass the glucose from that organ cells to the blood. Yes?
The adipocytes, the adipose tissue.
No, adipose tissue break down the lipids, triglycerides into fatty acids, right? And then fatty acid come out. So that is classically lipolysis. Any other tissue which is more actively involved in gluconeogenesis? Before you tell me something very unusual, it is kidney, proximal convoluted tubular cells. Just remember right now that kidneys can also do a small degree of gluconeogenesis. Is that right? So liver and kidneys both have glucose transporters type 2. Is that right? Then there is another cell in the body. There's another tissue in the body in which cells should have glucose transporters in two ways. Those cells don't produce glucose, but they want to have two-way glucose. Why? Because suppose that this is that cell. I will not tell you the name of the cell. You will guess it. These cells should have type 2 glucose transporters so that whenever glucose in the blood is high, it should immediately go into that cell. And if blood glucose level goes down, glucose should come out. And this cell loves to keep its glucose concentration equal to extracellular concentration. This cell is very, very sensitive to glucose level. What this cell is doing, whenever glucose level is going up, progressively glucose level in the cell is also going up. Whenever glucose level in the extracellular environment is going down, glucose level in this cell should also go in parallel down. Again, there's a very special class of cells in the body, and you know them, I'm very sure. And those cells love to keep intracellular level of glucose absolutely same as extracellular level of glucose, right? For that is why those cells have what type of transporters? Glucose transporters 2. And in glucose, whenever it's going up, in the cell also it goes up. And if glucose in the blood is going down, in the cell it will also go down. Who is going to tell me the name of these cells? Why these cells are so desperate to really keep their level of glucose equal to the blood glucose? Patrick. Okay, Gary. Carlos. Yes, Sean. No. Anyone? Okay, you tell me one thing. Which cells in the body, attention please, which cells in the body want to be extremely sensitive to the presence of glucose? These are beta cells of pancreas, because these cells have to release insulin in a very mild way. So these cells should be very, very sensitive that what is the level of glucose outside. So these cells should taste the glucose and then release insulin. So they should all the time sense the glucose. Of course, if these cells, beta cells of pancreas, if they are not permeable to glucose, can they know when to release insulin and when not to release insulin? That is so simple. So beta cells of pancreas, they are, look, if I say that you should be sensitive, for example, you are the insulin releaser. Let's suppose, come here Rahil, just come here. Let's suppose he is our beta cell of pancreas, right? This, oh my God, this beta cell of pancreas, right? The main function of this is to sense the glucose and release the insulin. So glucose has to go inside the cell, and then intracellular mechanism determine the level of glucose in the blood, and according to that they release insulin. How exactly molecular mechanism operate, we'll discuss in insulin. So beta cells of the pancreas are very, very sensitive to the blood glucose level. Is that right? For that purpose, they should be freely permeable to blood glucose, and only one transporter allows the cells to be freely permeable to glucose, that is type 2 transporter. Because let's suppose glucose level in the blood goes high, lot of glucose goes in and it starts secreting insulin. Insulin help in utilization of glucose, and now blood level of glucose goes down. The glucose which was previously in, that should come out, so that cell should know what is the exact level of glucose in the extracellular environment. So how the beta cells of pancreas know the extracellular level of glucose? Answer is, beta cells of the pancreas are always equalizing their internal glucose concentration with extracellular glucose concentration through which pathways? Which glucose transporters? Type 2. So if I ask you that what are the glucose transporters, what are the tissues which should have two-way glucose transporters? Number one, those tissue which want to release the glucose in emergency, like liver and kidney, which are involved in gluconeogenesis. That in emergency situation, when your blood glucose level goes up, liver start doing gluconeogenesis, kidneys start doing gluconeogenesis. Of course, they want to release this glucose into blood. So those two tissues plus the third tissue is which one? Beta cells of the pancreas. So we know type 1 and type 3 which are present in most tissues, especially in RBCs and brain. Type 2 are in the liver, kidney and pancreas, beta cells of pancreas. Type 4 are insulin dependent glucose transporters. They are specifically present in those tissues which want to store glucose, right? And glucose storage is especially in muscle. Glucose can be going to muscles and transferred into glycogen. And second is adipocyte. Now, glucose transporter 5. Glucose transporters 5. Who will tell me what a special thing about glucose transporters 5? First thing is, they are not glucose transporters. They are fructose transporters. They are fructose transporters. So glucose transporter 5 is primarily a fructose transporter. Right? Now, which are the tissues which have to transport fructose? Number one should be GIT cells, gastrointestinal cells, because in the GIT, you know, from the digestion of carbohydrate, glucose is produced, fructose is produced, and galactose is produced. So of course, these are the mucosal cells. From here, glucose should go into blood. Fructose has also, and galactose also need to be. So there are special fructose transporters which can take fructose from the GIT lumen to the cells. And there is one more tissue. Fructose is more sweet or glucose is more sweet? Fructose is more sweet or glucose is more sweet? Fructose is more sweet. That is the right answer. Right. So, number one, in the GIT there must be type 5 glucose transporters which should help in transport of fructose. Fructose is very sweet, and there is some organ in your body which loves the sweet things, especially that loves fructose. And that organ has cells which have transporter 5 because that organ cells love sweet fructose. So which organ is present in your body, Patrick, which loves sweet things?
My whole body.
Really, when I will... No, he thinks his whole body is like that organ and loves the sweet thing. This, no. Okay, first Patrick is saying, my question was that, which organ in your body loves to use fructose and needs glucose transporter 5, right? I asked the question to all of you. Patrick was very ready to say his whole body like to have fructose transporters, sweet things, right? Not liver, something really which loves the sweet things. You don't have a concept of sweet things. I think, who should be able to tell? Robert should be able to tell. Something, some organ in your body, I'm sure. You have a pair of those organs. Please tell me testes. Testes cells love to run their metabolism on fructose. Yeah, they love the sweet fructose. And I don't know, Patrick was saying I'm all like that. I don't know. Right, but anyway, we should not concentrate on Patrick's statement. Right? So, let's come back. Right? Don't think of Patrick a lot. Right? So, testes love to use fructose. You get worried. Why? Why? I will tell you later on, right? So right now you just believe me, testes love the sweet things, including fructose, right? And fructose should be transported to testicular cells. Is that right? Okay, I will tell you one more cell which love fructose and sweet things. Now you have to be very wise and intelligent to guess it. There is one more cell which loves to run its activity on fructose. Sperm. You don't know sperm? Sperm. Fructose. They love the sweet things. You should know by this age, right? So sperm love the very sweet monosaccharide and run their metabolism on that, and that is fructose. But anyway, right now we are not talking about sperm, and we are just talking about type 5 glucose transporters, which are in fact fructose transporters. And number one, GIT is having fructose after digestion of many polysaccharides, so it has to transfer that fructose to where? To the blood. So it needs type 5 glucose transporters. And once from the GIT, fructose goes to liver, most of fructose is converted into glucose. But little fructose still comes into blood, and that fructose will be utilized by your testes only, right? Okay, so this was something about glucose transporters. Type 7. Type 7 transporter. Okay, before you tell me very funny things, right? Type 7 glucose, just a minute. Type 7 glucose transporters are present in endoplasmic reticulum. Endoplasmic reticulum is network of tubules within the cells. And these networks of tubules have glucose transporter 7. Actually, endoplasmic reticulum is involved in gluconeogenesis. Endoplasmic reticulum is a network of tubules in the cells, right? When we say that liver cell can do gluconeogenesis, and we say that in the kidney also some cells can do gluconeogenesis, the site of gluconeogenesis is endoplasmic reticulum. So they should have some transporter for glucose, right? And they are having a very special type of glucose transporter which is called glucose transporter 7. Yes, please, you have a question.
Is it both
No, smooth is specially interested in gluconeogenesis. Rough endoplasmic reticulum is more concerned with protein synthesis because rough endoplasmic reticulum has ribosomes on it, which are the protein synthesizing organelles, right? Let's have a break now.